A flawless 5-carat diamond might run you $50,000 on the wholesale market, but a single cubic millimeter of a protein crystal, a microscopic speck weighing roughly 1 milligram, can cost $500,000 to produce. Before we can use X-ray crystallography to map 3D biological structures or design targeted drug therapies, we must first lock these proteins into a stable crystalline form. This tiny speck is the physical requirement for seeing life at the atomic scale. Growing one involves an intense physical contradiction. Mineral crystals, like salt or diamonds, are made of simple, rigid building blocks. Proteins are massive, flexible polyions. You are attempting to coerce thousands of squishy, irregularly shaped blobs to stack themselves into a perfectly rigid brick wall. Because every protein has a unique, irregular surface and a complex distribution of charges, there is no known method to predict exactly which chemical environment will cause a specific molecule to crystallize. The sheer number of variables has led some practitioners to view the process as a matter of luck or mystery, relying on anecdotal evidence and commercial screening kits to find a result. But relying on chance is not a viable strategy for a $500,000 pursuit. To reliably force these molecules into order, researchers have to move past superstition and master the physical chemistry of thermodynamics. The primary method is the hanging drop vapor diffusion technique. A researcher mixes a droplet of purified protein solution with a precipitation cocktail on a glass slide. The slide is flipped and sealed over a reservoir with a higher concentration of reagents, creating an isolated microenvironment. Because the drop is less concentrated than the pool below it, water vapor naturally leaves the drop to equilibrate with the reservoir. As the water evaporates, the droplet shrinks, squeezing the internal components together. As the water volume decreases, the protein exceeds its standard solubility limit. The drop enters a highly concentrated metastable state called supersaturation. We have now coaxed the proteins out of their stable solution, setting a thermodynamic trap. But even in this crowded state, the molecules still require a specific physical trigger to begin self-assembly. This graph illustrates the kinetic barrier the molecules must overcome. In this supersaturated zone, proteins are constantly colliding. To form a viable seed, a process called nucleation, they must collide in the exact right orientation to lock together. Most of these collisions fail. They form transient, unstable clusters that immediately break apart in the liquid before they can reach a critical, stable size. The physics of the system is working against them. Taking freely tumbling, independent proteins and locking them into a rigid lattice causes an unfavorable loss of motional entropy, which the system naturally resists. The energy required to overcome this resistance comes from the clathrate cage, a shell of highly ordered water molecules trapped against the surface of every protein. When two proteins finally lock together correctly, their surfaces overlap. This interaction immediately displaces and releases those trapped water molecules into the surrounding liquid. Freeing dozens of ordered water molecules creates a massive gain in entropy for the solvent. This gain is large enough to outweigh the entropy lost by the proteins as they lock into place, driving the entire crystallization reaction forward. Because the molecules only connect at a few sparse interaction points, the final product is not a dense solid. It is an incredibly fragile lattice, consisting mostly of empty voids filled with solvent. Here we see lysozyme crystals of varying quality. To the naked eye, a sharp, isolated block looks like a success, while intergrown clusters look like failures. However, external appearance is not a reliable indicator of data quality. Internal flaws often form during growth. If contaminants enter the lattice or growth happens too quickly, new layers of molecules can misalign. This creates distinct, slightly disjointed domains within the crystal, a condition called twinning or high mosaicity. This creates a laboratory paradox. Visually flawless crystals often yield useless, overlapping X-ray data, while an ugly, broken shard might provide a perfectly clear structural map. Simply swapping out chemical additives usually fails to correct these deeply internal structural errors. The environment can only do so much to fix a molecule that is resisting order. If a protein is physically averse to periodic packing, no amount of environmental tweaking will produce a high-quality crystal. Success relies entirely on these specific microscopic anchors, often charged amino acids or flexible loop segments, bridging the gap between the larger ribbon structures to form crystal contacts. When a protein lacks these connection points, researchers use protein engineering. They rewrite the genetic code to truncate flexible tails or mutate specific surface amino acids to forcibly create favorable contact points. Instead of blindly screening thousands of chemical cocktails, researchers instead spend their resources modifying the biological specimen itself until it is physically capable of packing together. Every atomic-level map of a biological process and every structure-based drug relies on succeeding at this specific thermodynamic game. Mapping the building blocks of life requires moving past the idea of magic and systematically coercing chaotic biology into the uncompromising order of a $500,000 crystal. The human body runs on machines that are entirely chaotic. Inside our cells, proteins are constantly wiggling, folding, and morphing in a fluid environment. Membrane proteins, for example, act as the primary receptors for most modern drugs. To design better medicines, researchers need an exact map of their three-dimensional structures, but you cannot measure something that refuses to hold still. The solution is to force these dynamic, squishy molecules to lock together into perfectly ordered, repeating grids, a state we know as a crystal. The difficulty is that proteins are fragile. They naturally resist being packed into a rigid structure. If you push the protein too hard or too fast to solidify, it fails. The structure collapses into useless liquid oils or solid precipitates. Even under highly controlled laboratory conditions, the chance of yielding a usable, diffracting crystal hovers right around 10 to 20%. You can't just drop the temperature and freeze the water like an ice cube. Researchers have to coax the protein into a delicate, temporary balance known as a metastable state. To find that exact state, they rely on an invisible thermodynamic map called the phase diagram. Understanding the boundaries on this map is the only way to beat the astronomical odds of the molecular casino and get the protein to perfectly self-assemble. The primary vehicle used to transport the protein onto this map is a process called vapor diffusion. In the classic hanging drop technique, a researcher mixes a 1-to-1 ratio of liquid protein and a chemical precipitant. They suspend this single drop upside down over a reservoir of a saltier precipitant solution. This phase diagram tracks the protein's state. The drop starts here, safe, but too weak to crystallize. As water vapor diffuses into the reservoir, the drop shrinks, pushing the solution across a thermodynamic threshold into a supersaturated state. To start, concentration must increase until the protein reaches the spontaneous nucleation zone, where the very first crystalline seed can form. But this zone is dangerous. If evaporation happens too quickly and the drop pushes too deep into supersaturation, the experiment fails entirely. If the evaporation pushes too far, chaos takes over. All the available protein is instantly consumed in a sudden shower, creating thousands of microscopic crystals that are entirely useless for data collection. The ideal path stops at the nucleation zone's edge, pausing just long enough for a single nucleus to form. As it grows, it absorbs dissolved protein, lowering the drop's concentration and returning the system to a stable equilibrium. By precisely navigating this exact sequence of evaporation and equilibrium, scientists can coax chaotic biology into mathematical perfection. The final challenge is that no two proteins share the same phase diagram. Variables like pH, ambient temperature, and specific amino acid sequences alter the boundaries of the maze every single time. Because the map is always shifting, researchers cannot rely on manual, single drops to find the correct path. Modern high-throughput laboratories rely on robotics to test hundreds of different chemical cocktails simultaneously. These automated systems operate with extreme precision, dispensing drops as small as 100 nanoliters into massive multi-well plates. We aren't taking a single guess at the maze. We deploy thousands of automated probes all at once to find the one exact path that yields a crystal. Even when everything goes perfectly and a large single crystal emerges, it remains incredibly delicate and sensitive to the slightest environmental shift. Researchers carefully harvest the crystal, bathe it in a protective chemical cocktail, and flash cool it to cryogenic temperatures. This deep freeze protects the sample from rapid radiation damage, allowing the crystal to survive being blasted by high-energy X-rays to read its atomic structure. Every precise three-dimensional model of a virus, a DNA complex, or a life-saving drug receptor we have today exists for one reason. Scientists successfully guided a microscopic drop of water through an invisible thermodynamic maze.